Welcome everyone to this virtual journal club hosted by the Physiological Society. Today we're going to be discussing a paper by Dr. Mike Urbin and his colleagues, Corticospinal recruitment of spinal motor neurons in human stroke survivors. So I'm Ainslie Johnstone. I am a postdoc at UCL. I work in the lab of Professor Sven Desman and Professor Nick Ward, and we're looking at using non-invasive brain stimulation or pharmacological interventions to try and improve plasticity in humans. And today I'm joined with two other panelists, Dr. Mike Urbin, who is the author of this paper that we're going to be discussing today. Mike is jointly affiliated with the Rehabilitation and Neural Engineering Lab at University of Pittsburgh and the Human Engineering Research Labs at the VA Pittsburgh Healthcare System. He's primarily interested in human motor control and motor learning and uses a variety of non-invasive stimulation and structural imaging methods to study physiology of stroke and spinal cord injury. So welcome, Mike. And we also have here with us Professor Charlotte Stagg, who is a professor of human physiology at University of Oxford and the head of the Physiological Neuroimaging Group. She is interested in how the brain adapts to new challenges, particularly focusing on the physiological processes underlying learning new motor skills and also recovery after brain injury. Her work spans across modalities from MRI and MEG to non-invasive brain stimulation and pharmacological interventions, and recently has started spanning across species as well. So initially focusing on humans and now moving into some rodent work as well. Charlie's an editor at Journal of Physiology, and also she was my PhD supervisor. Most important accolade. Okay. So welcome to you all. Thank you for joining. I'm just going to quickly run through the format for the session today. So this section of the session is the virtual journal club. This will last for about 45 minutes. Well, first of all, do a little bit of an introduction of the panelists, already done that, and the paper. We'll then move on to Mike, who will give us a bit more details about the rationale, methods, and the results from his point of view as an author. And finally, Charlie will tell us about what made this paper publishable in the Journal of Physiology and any insights that she has from her perspective as the editor. We'll then have time at the end for Q&A. Hopefully we'll have a good half an hour for that. So please do submit your questions. You can submit your questions using the chat function here. You can do it anonymously, or you can say who you are. It would be nice if you can let us know who you are and also let us know where you're coming from as well. If you see other questions in the Q&A that you like the look of, or if you also had that question yourself, you can choose to upvote the question and it will move to the top of the queue and we'll be more likely to get to it. But also don't worry if we don't have time to answer your question or we run out of time. Immediately after this session, there will be a networking session, which will be much more informal. Everyone can chat and we can get to any questions that might have been missed. So there'll be a link provided for this networking session at the end of this current journal club session, so people can move over into that session there. Just a reminder, this meeting is recorded and you'll be emailed the recording in about a week's time, as well as a feedback form to give us your thoughts on the session. One last thing before we get going. You can get involved with the Journal of Physiology in a lot of different ways. You can host a future meeting of the virtual journal club like this one today, if that's something you're interested in, and talk about a paper that you've recently read in Journal of Physiology and get to speak to the authors about it. You could also write a journal club article. I know a few of my colleagues who've done this. It sounds like quite a fun process, just a short article about another Journal of Physiology article. Or of course, you can submit your research. So Journal of Physiology publishes original research in all aspects of physiology. You can find all this information on the website, or you can get in touch with these email addresses. Okay, so just very briefly before I hand over to Mike to give the details about the paper, I just wanted to talk about why I chose this paper and why I thought it was interesting. So to me, the first thing I thought of when I read this paper was that it was really taking a bit of a different approach or a different way of thinking about stroke recovery to what how I was normally used to thinking about it. So to me, when I was previously thinking about stroke recovery, I would always be thinking about what was happening in the brain. So stroke has caused some damage to the brain. How can we alter the brain in some way to try and fix motor problems or help with motor problems that these patients are suffering? And I think oftentimes I would be completely ignoring or just completely forgetting that there was a whole motor system that was downstream of this brain that I was so focused on. There's the spinal cord, and of course there are the motor neurons, which in the case of stroke are pretty much intact. And it was this that I really thought was something that I found very interesting about Mike's paper, is that what they were doing is really looking at this sort of intersection from the brain, which obviously has some kind of damage caused by the stroke, and the rest of the motor system that should be functioning as normal. So just very briefly to talk a little bit about the rationale before Mike goes into some of the details and a little bit about what they found, just as a little teaser. So the rationale, as I understood it, was that after a stroke, these downstream motor neurons are pretty much intact, even years after the stroke, years after they have essentially been deafferented from the downstream, or deafferented from the brain. But this damage to the brain will be causing a reduction in the recruitment of these motor neurons, in part just because the brain pathways that would be previously connecting with these neurons are now damaged. So if this is the case, perhaps it's possible to improve the recruitment of the healthy but effectively cut off motor neurons from this residual corticospinal tract. So the paper had two main questions. Do stroke patients have reduced recruitment of motor neurons? And if so, can this recruitment be improved? Just to do a little spoiler before Mike takes over, answers to those questions were yes and yes. Mike, do you want to give a little bit more detail?
I'll unmute myself first, and then I'll go ahead and share.
All right.
Okay, you're able to see that? Okay, very good. So thank you for the introduction, Ainslie, and thank you everyone for your attendance today. For the interest of time, there's two main areas that I'm going to briefly cover in the slides that follow, and they mainly address the motivation for the study and then also an explanation of the triple stimulation technique. The hope is that this information will help guide discussion. So regarding motivation, we know that there are multiple neurological mechanisms altered by stroke that contribute to movement dysfunction. Teasing apart the contributions of all these mechanisms is complicated by the fact that heterogeneity in stroke location exists. There's also widespread brain regions that are impacted due to diascosis and other factors. In general, however, distal limb weakness has a great deal to do with mechanisms further downstream in the spinal cord. Specifically, strokes causing corticospinal damage lead to abnormal interactions between upper and lower motor neurons. The neuromuscular junction, in fact, is one of the most reliable synapses in the central nervous system. So the problem really of paresis exists in the inability of cortical motor areas to activate spinal motor neurons. So with regard to human stroke survivors, for example, we know that from tractography analyses that white matter integrity of the corticospinal tract is linked to functional capacity and is a fairly strong predictor of motor recovery. We also know that motor evoked potentials elicited by magnetic stimulation of primary motor cortex are reduced in size and prolonged in latency, as shown on the slide in a couple of survivors who have participated in my studies. However, these metrics do not directly capture how well or how much of the spinal motor neuron pool is activated. This consideration in part led to the current study's purpose, which was to characterize corticospinal recruitment of spinal motor neurons in humans with long-standing motor impairment secondary to stroke. So how is this accomplished? Well, a non-invasive electrophysiology method was developed about 20 years ago and became known as the triple stimulation technique. Despite gaining traction early on, administering it presents some technical and practical challenges, which is why I suspect not many studies have been published, particularly in clinical populations. But in order to appreciate the information gained from the method, it's helpful to understand how it works on a conceptual level. So I've tried to break it down briefly in the slides that follow. The first thing to understand is that this triple stimulation method is a collision technique. As its name suggests, stimulation is applied to three neural elements at set intervals, or delays calculated with the simple equation shown here. These stimulation pulses result in two potential collisions occurring in the periphery distal to the spinal motor neurons. To complicate things a bit, there are also two different waveforms generated depending on which elements are stimulated. These waveforms, referred to as test and control curves, are shown on the right. The control curve, which is shown in blue, is essentially a normalization for the test curve. So one aspect of my job that I very much enjoy is being able to help stroke survivors who participate in my projects develop a better understanding of the reasons for their impairments. And since this triple stimulation method is a bit complicated, and as Ainslie put it quite nicely, requires some mental gymnastics, I've come up with a way of explaining the method that appears to make some sense even to individuals without backgrounds in neuroscience. Hopefully it will also prove useful today. So the method is a collision technique, and therefore the sport of American football offers a pretty useful analogy. Now, acknowledging the international audience in attendance today, I want to make sure that I make clear I make no claim that American football is any better than the sport of European football, which I acknowledge is an important distinction to make. Anyway, to reiterate, the technique involves three stimulations eliciting separate volleys, which you might think of as three different position players in the sport of American football. The first player is the volley from stimulation to the brachial plexus at Erb's point, which we can think of as a blocker on offense, whose job it is to thwart defenders from tackling the ball carrier. The second player is the antidromic volley from stimulation to the ulnar nerve, which can be thought of as the defender, whose job it is to tackle the ball carrier. The third player is the ball carrier, which results from a second stimulation to brachial plexus. As you may already know, the objective for the ball carrier is to cross the goal line with the help of the offensive blocker, resulting in a control curve, as shown in the waveform to the right. An important point to make here is that stimulation is supramaximal, meaning that all axons under the stimulating electrode will be depolarized. That said, there should always be one blocker for every defender, indicating that the ball carrier will always reach the goal line in the case of the control curve. It's also important to remember that the control curve serves as a normalization for the test curve in which the same three players are involved. The one key difference is that the blocker on offense now is elicited by stimulation to cortex instead of the plexus. Accordingly, you'll notice that the blocker is positioned presynaptically to the alpha motor neuron. However, just as the control curve, the defender and ball carrier come from stimulation to the nerve and plexus respectively. Objectives for all players are still the same, but the key distinction is that cortical stimulation will not always recruit all spinal motor neurons because of potentials for failure in conduction along the tract and in turn recruitment. Nevertheless, superimposing the test curve on the control curve provides an index of how many axons or how much of the spinal motor neuron pool is recruited by corticospinal inputs. In essence, the greater the number of collisions, the greater the number of motor neurons that will be recruited. So let's consider two hypothetical cases of test curves to illustrate my analogy a little bit better. In both cases, let's pretend that 4 motor neurons represent the entirety of the motor neuron pool innervating a target muscle. In this particular case, an intrinsic hand muscle, the first dorsal interosseous, or FDI. In case A, 3 blockers are recruited, creating collisions on 75% of axons, allowing 3 ball carriers to reach the goal line. The result would be a 75% superimposition on the control curve. Stated another way, you can think of 75% of the motor pool has been recruited. In case B, however, 1 blocker is recruited, creating collisions on 25% of axons, thus allowing only 1 ball carrier to reach the goal line. The result would be a 25% superimposition on the control curve. So because more blockers were recruited in case A, there will be a higher test control curve ratio reflecting greater recruitment. So I acknowledge that the analogy is imperfect, but it illustrates how collisions are taking place in this context to quantify corticospinal recruitment of spinal motor neurons. So looking at some actual data here, we have two individuals, two stroke survivors who underwent testing using the technique. The individual on the left is less impaired with some residual movement in the paretic FDI, and able to apply movement against resistance as well. The individual on the right, however, only has a trace level of contraction. So in other words, you can record EMG, but there's really no active range of motion, just a very feeble contraction that you can observe in the muscle. When I designed the experiments, I included two cortical stimulation intensities based on the hypothesis that recruitment should modulate with the intensity of cortical stimulation. So shown here are test curves with low stimulation intensity, or what I defined as 1.5 times resting motor threshold. And then superimposed above those are test curves with a higher stimulation intensity at 1.7 times resting threshold. It may not be entirely easy to see, but recruitment does modulate with stim intensity in these survivors. More importantly, you can see a rather significant disparity between subjects, with the more impaired individual showing a much lower test control curve ratio. Another consideration that's really important when we do these type of experiments that involve non-invasive techniques is how we can be certain that we are capturing the phenomenon of interest. In this particular case, how can we be sure that collisions are actually taking place? As many of you likely know, the compound muscle action potential, or M waves, at supermaximal stimulation amplitudes are very consistent in both their size and their shape. In the case of test curves, however, you'll notice that the size of the M wave is reduced considerably, particularly in the case of the more impaired survivor. And again, looking at the test control curve all the way on the right, that is a CMAP, that's an Mmax, and for it to be reduced like that suggests to me quite strongly that there are collisions taking place and that the technique is providing us the information that we want. So that's the information that I'm going to provide on the triple stimulation technique. Ainslie, I didn't know if there was additional information you were going to be providing for results or if there was other discussion. Are you muted?
Yes, sorry, I am. Great, yeah, I mean, I don't know, perhaps we want to go over we can just verbally, I suppose, go over some of the results in a bit more detail of your findings, perhaps. Or I don't know or if people already have questions.
So, I mean, I can go ahead and hit the high points. The first part was that, we could actually show that the extent of recruitment was providing some type of meaningful information about, an individual's movement abilities. And so, we essentially, based on how we conceive of the experiments, chose to use, a clinical test of, muscle activation, simple medical research council for muscle strength. But then we also looked at pinch force as well. We tried to do a lot, and this was all happening during the pandemic. And so unfortunately, we weren't able to get measurements of dexterity, but those experiments are ongoing. So, we're also going to be looking at whether or not this, this biomarker, so to speak, provides useful information about motor neuron recruitment. But the key point to remember is that, we typically index changes in white matter integrity, latency of motor evoked potentials, which all provide critical information. But in this particular study, we saw that the latency of the motor evoked potential was not related to their ability to produce a pinch force. It was related to recruitment in general. So having established that, we then started to look at this idea of trying to reverse the failure in recruitment to see if that was something that was malleable. And so there's this neuromodulatory technique that's been around for a little while now. It was originally showed about 10 years ago, and then was then taken into spinal cord injury. And it's this idea of paired corticomotor neuronal stimulation. And what that idea is that you're basically targeting the synapse between upper motor neurons and lower motor neurons to improve synaptic connectivity. And that's via principles of spike timing dependent plasticity. And what we were able to see is that in a good number of the survivors, and it was generally the people that were more moderately impaired, we could actually see changes in recruitment from this neuromodulation protocol that takes all of 15 or 20 minutes to administer. So it provides some evidence that we have these changes in recruitment that manifest as a result of stroke, but then there's potential to potentially reduce the reduction in recruitment that you actually see.
Great. Charlie, do you maybe just want to discuss quickly why you thought the paper was publishable in Journal of Physiology, and then maybe we can move on to some questions?
Yeah, definitely.
I have a few.
Thank you both. That was a really nice summary of the paper. So yeah, I'll go very briefly through kind of what happens when a manuscript gets submitted to the journal. I know some of you will know this, but you know, it's one of those things that only becomes obvious after a while, I think. So every time you submit a manuscript, it will go to the senior editor, and who these people are is not a secret. If you look on the papers, if you look on the papers, it will say who the editors are. Okay, so you can get a feel for, you know, who the people are who are going to be looking at these things. In this case, it went to Richard, who's the senior editor for neuroscience. He had a look at it, he'd read it quite carefully and worked out which of the deputy editors sort of remit it fell under. And my remit really is around motor neurophysiology, and particularly, you know, in stroke recovery. It's an interest close to my heart, so it got sent on to me. At that point, the deputy editor will read it again very carefully. We triage reject, so at the point we do it, it will get rejected. About a third to a half of papers that get submitted to the journal gets rejected at that point. The things that we're looking at to make it through that stage are that it fits into the journal scope. So it really is physiology. You know, we are the Journal of Physiology and we care about basic physiology. And this paper obviously spoke very well to that. It needs to be quite clear what the advances are. You know, we like, you know, we take papers where there is a clear advance in understanding. And that has to be, you know, that's a difficult thing to quantify, and it's quite a subjective determination, but you know, you need to be very clear as to why this paper, you know, really tells us something we didn't know before, and why the readership would care about that. And so, you know, Mike's paper did that very nicely, and it's very clearly actually the covering letter and in how the paper is written, which was very clearly laid out and very logical, took us through this. It was very clear what the problem was, why it was an interesting problem, why we should care about it, how they answered it, and then, you know, what effect that had and why that might be useful. So it very clearly did a lot of the work for us, and really became very obvious that it was definitely something within the scope and within the interest. The other thing that, you know, I think it is worth saying is that, as Mike and Ainslie both said, you know, it is a complicated paper, right? You used a difficult technique, but it was a difficult technique, but we can handle that. You know, we have hardcore physiologists, we have good reviewers, so don't be put off from doing things that are complex. But if they're complicated, you need to explain them. All right. And that was one of the real strengths of this paper, as you've just heard Mike do. You know, it is a complicated technique. It takes a while to get your head around. The figures really helped. There's a simple explanation of why it does it, what it does, why, you know, why that captures the information. So that was the really big hurdle. So if you've got a complicated technique, that is completely fine, but make sure you explain it to people who don't spend their lives doing this. So yes, and the other thing that we liked about it is not essential, but it was very clear that it's a physiology paper, but it has clear clinical relevance. And that makes it, that translation is something that is lovely if it's there. If it isn't there, it doesn't matter. We are a basic physiology journal and we take basic physiology papers. But in this case, it was very clear that there is a translational relevance to it. And that always adds a different perspective to the paper. So those are the things we liked about it. It got sent out, so we sent it out to review, obviously, because we're here. It got reviewed by 2 expert reviewers. I obviously won't reveal their identities, but one of the good signs of this paper as an editor is that they both accepted very quickly, and it was very easy to find people who wanted to review it. But we start getting alarm bells when it's difficult to find reviewers. And in this case, it was very easy, partly again, you know, so Mike was helping himself. So he put on there some really sensible reviewer suggestions, which we may or may not have taken, but they were very sensible, very reasonable. Don't necessarily put the very senior people in the field. They sometimes will do it, but they're often very busy. But people who are likely to say yes in the right kind of labs is very helpful. And we also will send it for statistical or ethical review if we need to. We have dedicated editors to do that. I don't think we did for this. I don't think there was anything we worried about, but we often do. And that just gives us a bit of backup if there's some complicated stats in there. And I do that, I'm afraid, if there are complicated statistics, just to make sure that we've got the right thing. And then it went to review, and things that I liked about it were the things the reviewers liked about it. And we got 2 extremely positive reviews back. I think they were pretty robust. We don't give people an easy time. You know, the scores were high and the comments were good, but they were extensive, and there were issues and there were discussions to be had. But they broadly, the reviews were very positive. And then the responses, were very good. They were very helpful. They were very polite, very respectful. They did the things that they needed to do. They were very clear as to what they'd done and why and where it was in the manuscript. And so both the reviewers, it took as often, the journal does have robust reviewer policies, and so it had a couple of rounds, but it was a very friendly and hopefully very constructive discussion. And it was a great paper to start with. And I think Mike's a bit closer to it. I think it ended up a stronger paper after the revisions, which is what we would hope. And we're very pleased to have it. It's a very nice, clear paper that says something important about physiology that hopefully is of interest to a wide readership. So that's the kind of thing we're looking for.
Great. That's great. Thank you, Charlie. Great. So we have a question. We've got 2 questions now, actually. So I can read them aloud. So I think probably the first one is for you, Mike. So this is from Paul Ansell from Northumbria. He says, with the PCMS protocol, did you individualize the interstimulus intervals, or did you apply the same ISI for all participants? So perhaps it would be good to just do a very quick overview of that protocol for people who are not so aware of it.
Yeah. So no, it was individualized to make that clear up front. I don't know of many papers that have actually taken the approach of using PCMS at a standardized ISI. Most of the papers out there are individualized. And the way that we do it essentially is we look at various latencies, specifically M-wave latency, F-wave latency, and then the latency of the MEP with slight background activation. And we use those to basically calculate peripheral and central conduction times. And then once we have those conduction times calculated, we're essentially timing the presynaptic depolarization to occur 3 milliseconds before postsynaptic depolarization happens. So it is based on individual latencies, and the target is that there would be a 3 millisecond arrival of presynaptic volleys before postsynaptic volleys.
Great. Okay, we have another question again about the protocol from Ricardo Mesquita. He was wondering whether all participants responded similarly to the PCMS protocol, or whether there was some inter-individual variability. Could the level of response be influenced by the type of stroke, for example? So another one for you.
Yeah, absolutely, great question. And this is, one of the things that came up quite a bit in the reviews is that, we saw that the more moderately impaired individuals were more likely to show the response, which is consistent with what we've seen in spinal cord injury. And there's been some other work that's been done. I think just a single paper that's looked at, kind of a way around that. How do you get people that are more severely impaired to actually show the response? But for the purpose of this investigation, we essentially just put everybody through the same protocol, which was just a resting muscle, 100 pairs of pre-input of paired stimuli, and then just measuring the response at various time points. And so what I can say is that the people that didn't show the effect tended to be the people that really had trace levels of activation. And if we think more about how TMS works, right? There's a series of descending volleys, and the summation of those volleys is what leads to presynaptic activation. And so when we look at evidence from spinal cord injury, what we know is that the periodicity of those volleys it's spread out. It's not coordinated, and so those inputs to the spinal motor neurons don't summate properly, and that leads to variability in activation. And so it's quite possible that might have been part of the reason why we didn't see that in those individuals. I did show some tractography data from some of the participants actually in the study, and you can see that there's some pretty significant severing of the corticospinal tract. You have very few fibers that are conveying the remaining signal. And so it was relatively unsurprising that was the response, but we are currently working through additional experiments to see if we can find a way to mediate the effect in individuals that have those high level impairments.
Yeah.
The last part of the question is, how it's influenced by type of stroke. So I mean, when we think about type of stroke, I think about location. I think about ischemia versus hemorrhage. This was a very heterogeneous population that we had, or a heterogeneous sample that we had rather, which reflects the population at large. And so I could envision a scenario where that might matter. We do have structural imaging on these individuals, so in some of the individuals, so we'll be taking a look at that further. But that is a good question.
Great. So just to sort of go over that just to clarify from my point of view. So what you're saying is that in the patients who perhaps had more damage, this paired stimulation, where the idea was that the volleys would be sort of meeting at the correct time to sort of strengthen that synapse to try and improve the recruitment, that was just maybe not quite happening exactly sort of timed perfectly in those patients who had the most severe impairments, the stimulation on the cortex and the stimulation on the nerve.
Correct. Yeah, I would think that basically, the timing of those, the I waves and D waves at the level of the spinal motor neurons just basically didn't summate properly, and therefore you didn't get a complete activation. And so this whole idea of spike timing dependent plasticity really, couldn't exert its full effects because the timing component was probably not, appropriate. We did use, their individual latencies, but as we know, there's a lot of variability in those responses, and so that's just a rough approximation on my end in terms of interpretation. But like I said, additional experiments are underway.
Okay. I'm just going to read over this next question before I read it out, but just quickly on that, I was wondering if you had any comments about what that means or what this whole study really means for sort of clinical practice, like where you see this going, what you feel needs to be done before, if this is could potentially become a useful technique for patients.
Right. So I'm a skepticist by nature, and so I think that there's a lot of hoops that need to be jumped through just on the logistical end of things, right? And it's different all over the world, but in terms of in the States, there are limitations in terms of how much time individuals actually get in terms of physical therapy, occupational therapy, what have you, and presumably those would be the forums where this type of treatment would be administered. And so, there is a setup involved, there's that cost, but then you're also taking time away from, motor retraining potentially to be able to actually administer the protocol. So there's those type of things that you always have to kind of be mindful about in terms of, how this would be applied clinically. But more generally, I think that, the idea is that if you can actually enhance movement potential, right? That's what this PCMS protocol has been shown to do, is that it enhances, in this case, what we've seen is recruitment. It enhances corticospinal transmission. People have shown effects on voluntary motor output. So the idea would be that if you have the ability to better produce movement, then you may be able to obtain better therapeutic benefit from the treatment that you are receiving. Great.
Okay, we have a question from Baman Nasserol-Salmi. Sorry, apologies of my very poor pronunciation. They'd like to know, again, have a little short explanation of what the triple stimulation technique is providing over and above CMAPs and paired or single pulse TMS. What is it really, what is its sort of insights? Again,
right.
for you, Mike.
Right. Yeah, that's an interesting question, and it's kind of nuanced. So when we think about what this technique is doing, you really have to buy into the fact that these collisions are taking place where they're taking and you're capturing corticospinal recruitment of spinal motor neurons. And so when you look at for example, normalizing your MEP max to a CMAP or some other type of measurement. I mean, this metric is unique in that it actually captures that. In the paper, I kind of draw on that distinction in that, people have looked at white matter integrity, people have looked at various metrics that correlate with recovery and function. But this is, actually telling you something unique, right? It's telling you about how well the primary motor cortex can activate the spinal motor neurons. So to that end, the insight, the information that you're gathering is, I would say, probably more direct as direct as it can be in the context of non-invasive stimulation. Because, we know from animal models that this phenomenon exists. It's just, it hasn't been really directly captured, I would say, in human stroke survivors. I think you're muted again,
Yeah,
Ainslie.
sorry. Okay. Sorry, we just had a question there from Sarah Hussein. She asked, she said, joining from Austin, Texas, can you speak to the tolerability of these techniques? So both the triple stimulation technique and the PCMS intervention in stroke patients who often have decreased pain thresholds.
Yeah, that's a great question. Urb's point stimulation, stimulating at the plexus is really what is a little bit less tolerable than the other techniques. And what I would say about that is the control curve in particular, because you're doing these stimulations in rapid succession, and as I mentioned during my 2 pulses and 2 supramaximal pulses to the plexus in very short order, on the order of milliseconds. And so that tends to be very difficult, but realistically, because of what it's providing, you only need to do 1 or 2 of those at baseline to really get a sense of what the control curve should look like. When it comes to the test curve, obviously there's only 1 stimulation applied to the brachial plexus. So what I would say is that there wasn't anybody who came into the study and thought it was fun, but at the same time, I did get people to come back for a second session, right? We did this on the paretic and non-paretic sides. And so, it was something where I think that, the people who participate in my studies understand that, we're working together to find new information, new physiological insights that can help people, recover from strokes or inform therapies, what have you. And so they're, very vested in the success of the project. And so I think that probably, gave them some motivation to want to be able to come back and do it a second time. But I would think if it was, just completely intolerable, this wouldn't be something that, people would come back for a second round. But, again, it varies from person to person. The vigorousness, or I guess the briskness of the response that you see, I think is really what bothers people. It's not necessarily a focal noxious pain that people are describing. It's more that their arm is really vigorously moving and they don't understand why, right? And I find that when you explain these things to them in layman's terms and help them understand what they're experiencing, they appreciate it a little bit better, and they kind of say, oh, well, now that I get that, this isn't really that bad. So I hope that answers the question.
Yeah, that's definitely interesting. What about, so in terms of the PCMS protocol, was that more tolerable for patients?
Yeah, I think that when you're talking about the PCMS protocol, right, it's magnetic stimulation to cortex paired with peripheral nerve stimulation, and so in that sense, most people have no issue with that. There's an initial exposure sometimes with electrical currents applied to nerves, but again, once they have some baseline familiarity with it, it tends to be pretty tolerable. The one thing I guess also that's challenging, and this is mainly a limitation of doing physiological experiments, is that you are taking a lot of measurements, and so some of these testing sessions can last upwards of 3 hours. And so really it's more so the sitting for that long that I think tends to be more problematic for people. Yeah.
I suppose the PCMS, that is if this was ever to be in some kind of clinical practice, that's the part I suppose that would be the most useful potentially. Do you have any thoughts on, I mean, I know that you said that you didn't have any, obviously these first sessions were very long and there was no time to collect any kind of functional measures, but do you have plans to look at whether the PCMS protocol is sort of improving the function through this increased recruitment?
Right, yeah, so what's been shown in individuals with spinal cord injury previously is that performance on a clinical scale of manual dexterity improves. What we're trying to do is we're actually trying to develop our own simulation to kind of more poignantly probe granular mechanisms of control. So like control processes when it comes to being able to perform dexterous behavior. So we have a simulation that we've been working on for about a year now, and we're in the process of actually starting to run those experiments. So the goal would be to kind of link the physiological information with performance information, but the performance information is a little bit more granular than what you would get with a clinical test. It tells you more about control processes that are influencing dexterity.
Okay, yeah, that's really
in the process.
Great. Charlie, do you have any questions you'd like to ask?
Yes, a couple, if I may. Just because normally editors don't get to. You can sneak a couple in sometimes, but normally you just have to, it seems a bit cheeky to get a third reviewer for free. So one kind of technical question, if I may, and then one more kind of big picture question. So just going back to Sarah's question about tolerability. So I've had M1 electrical stimulation. Now, I know you didn't do this to everybody, but you did do it to some people. It wasn't much fun. How did people find it, and what did you do to minimize the discomfort?
Right. Yeah, so there's two ways that you can approach the electrical stimulation. And just to provide a little bit of context for the audience, the goal for those experiments, they were control experiments, right? And we wanted to basically show that the PCMS was acting at the corticospinal motor neuronal synapse, right? So to be able to do that, there's really no good way to do that with magnetic stimulation. You might be able to indirectly do something with H reflexes people have talked about, but really the best way to do it is through electrical stimulation, either at the level of cortex or at the level of the cervical medullary junction, so producing CMEPs. And either one of those tend to be a little bit more difficult, and that depends on the current that we need to pump into you to be able to drive a response. And so the people that we tested were individuals that, first of all, were proven warriors in my lab that have been through a lot of the experiments and had exposure to all these things. So it's they came in knowing what they were getting into, and then we did kind of a familiarity thing where we gradually increased so that way they knew what was going on, and then certainly there's always the ability to stop at any time, but essentially that familiarity tends to help people tolerate. And then we only stimulate at the level that we need to drive a response. So I think we were targeting a certain percentage of Mmax. Once you get to that threshold, you can get those responses fairly reliably. And then of course we're only doing the minimum we need to be able to gauge any type of modulation that might be happening at a subcortical level, right? Because that's what the technique gives you, is a measurement of what's happening between cortex and the spinal cord. And so that gives you, this idea that there's some modulation happening in between. So it's one of those things where that in particular is not pleasant, but if you get people that have low enough thresholds that are very interested in the work, it tends to be, fairly straightforward. So.
Yeah, no, that's a good point. I mean, it's a very, very valuable technique. We should be really clear. I realized that it wasn't in my question, that we're not talking about transcranial direct current stimulation or alternating current stimulation or any of the neuromodulatory techniques, which are, a bit itchy, but fine. This is supra threshold electrical stimulation to induce MEPs, which is, there's a reason we moved on to TMS, except that it can, as you very eloquently said, it really is a very useful physiological tool. But yes, it's interesting to hear that, you thought quite carefully. Yeah, it's not something you do on everybody, right?
Great, we have two more questions in the chat. So I'll try and get through at least one of them, and then we can move over into the networking session and we can cover the any more. Okay, so Ronan Muniers is at the top. Have you tried measuring when the triple stimulation responses have you tried measuring the triple stimulation responses at the early acute or subacute stage when cortical hyperexcitability is most prominent? Would you expect this to show lower recruitment than at the chronic stage after cortical excitability has recovered, at least to some extent? And might this add further insights to the functionality of the corticospinal tract over and above the binary MEP presence or absence stratification?
Yeah. It's an interesting idea. If somebody, if we were able to get people to participate in a study longitudinally like that, I think that would provide, very useful information to exactly what Ronan is referring to. But, I don't know. I mean, again, teasing apart, what's happening in cortex from what's happening, at the level of the spinal cord is kind of different. But I would agree that, a longitudinal study like that would probably help you tease apart, an answer to that particular question.
Great. Okay, so I think we should maybe wrap that up here. There was another question from Ricardo, which I've copied and pasted into a Word document, so we can discuss that in the, or maybe he can just tell us it in the networking session. I think that Rosie is going to give us a link. Yeah, here we go. Link to the networking session is in the chat. So if you'd like to join the networking session, you can just click to follow that link, and we can all meet there very shortly, and you can ask your questions in person. We can continue the conversation. Thank you everybody for coming. This has been great.
Thank you.
See you over on another link.